Let's start. Assalamu alaikum. My name is Khalid Alnajjar. I will be your tutor for this lecture, Conversion of Amino Acids to Specialized Products. Before I start this here, right here is my contact information. You can contact me whenever you want, for whatever you want, regarding this lecture, the whole course, any other course. I'd be more than happy to help. Let's start. So I'll be talking about synthesis of different specialized products from amino acids. I will talk about using tyrosine as an amino acid to synthesize thyroid hormones, melanin, and catecholamines. This is one of the most important objectives of this lecture. And then using tryptophan to synthesize serotonin and histidine to synthesize histamine. It's wrong. And using arginine and glycine to synthesize creatine. And after explaining this, I will be talking about, the doctor added some slides about drugs that affect the process of the synthesis of those important products using those amino acids. طيب. So what's wrong with this? A quick overview or just a recap of what amino acids are used for. As you remember, amino acids are used for metabolic energy. I think I explained the lecture how to convert amino acids into glucose the previous PAL session. So yeah, we can use it as energy. We can also use it for protein synthesis. Protein is one of the most important molecules in our bodies. So amino acids are used to synthesize proteins, as you know. And as you remember, we don't have a storage for amino acids. There's a pool in the blood of free amino acids. Our body uses them whenever we want, or whenever the body requires them. We also use them to all of that before mentioned. I said before, all of those 2 points, have been explained before. For the last point of the last use of amino acids is synthesizing substrates or products, which will be the topic of this lecture and future lectures as well. Let's start with thyroid hormones, synthesizing thyroid hormones from tyrosine. Of course, this topic will be covered in much more details in EndoRepro next year, inshallah, a very interesting block. But basically, you only need to know that tyrosine, let me bring the laser point. Tyrosine is tyrosine produces thyroid hormones are derivatives of tyrosine. All of the process requires tyrosine to form thyroid hormones. We have two principal thyroid hormones, T4 and T3. Thyroxine is T4, triiodothyronine is T3. The doctor said you don't need to know the names, but it's better to know them. The only difference between T4 and T3 is the number of iodines or iodine molecules. So a thyroid hormone أصلاً is made up of tyrosine and iodine. T4 has 4 iodine molecules, and T3 or triiodothyronine has 3 iodine molecules. That's the only difference between them, literally. Melanin from tyrosine. Melanin, melanin is the pigment of our skin. It's synthesized from tyrosine in the melanocytes. Melanocytes are skin cells or cells in our skin layers that produce or synthesize melanin, which is the pigment, and it is there to protect our skin from harmful radiations from sunlight, for example. One of the examples, it is oxidized first to L-dopa and then eventually to melanin. You're using the enzyme tyrosinase. You can see here tyrosine. The whole process is carried out by tyrosinase. So tyrosine converted to dopa by tyrosinase, again to convert it to dopaquinone by tyrosinase as well, and then to melanin. One defect is albinism. You might know albinism. It's It's deficiency of the pigment melanin. Okay, it's usually a defect in the enzyme tyrosinase, since it's the only enzyme carrying out the function of or the process of synthesizing melanin. So albinism is usually a defect in tyrosinase, and tyrosine makes up melanin using tyrosinase. Okay, in the melanocytes. طيب. Now on to the main topic of this lecture, catecholamines. Catecholamines or catecholamines are epinephrine, norepinephrine, and dopamine, first of all. They are the fight or flight molecules that say so sympathetic nervous system. These are the molecules that in the sympathetic nervous system or fight or flight, basically, sympathetic nervous system. This is the whole process. There is a longer one I'll be explaining in the next slide, but it also requires tyrosine, dopa, dopamine, norepinephrine, epinephrine. This is a video. This is a video that the doctor added for if you want to know the functions of the sympathetic nervous system or fight or flight, or for example, if a lion is attacking you, what will happen? What are the physiological changes in your body that will happen accordingly? Sorry. This is the longer version of it. Let me explain it. So yes, it is synthesized from tyrosine, but before that, your body uses phenylalanine. But the main amino acid there is tyrosine. So phenylalanine is converted to tyrosine using phenylalanine hydroxylase, and this requires tetrahydrobiopterin, which is BH4. You will see it in the next slide, inshallah. Then tyrosine is converted to L-dopa by tyrosine hydroxylase now, which also requires BH4 or tetrahydrobiopterin. L-dopa is the first catecholamine in our process. L-dopa will be converted to dopamine, another catecholamine. Dopamine will be converted to norepinephrine using the enzyme dopamine beta hydroxylase. This step requires vitamin C and copper, as I'll be explaining in the coming slides. And the norepinephrine in the adrenal medulla will be converted into epinephrine. Okay, so your body first synthesizes norepinephrine, then converts it to epinephrine. Those are the catecholamines in our bodies. This slide just basically explains what I explained. Let's go over them quickly. Norepinephrine, dopamine, and epinephrine are catecholamines. They are synthesized. Yeah, dopamine and norepinephrine are synthesized in the brain. Norepinephrine is also synthesized in the adrenal medulla and then gives off epinephrine. So dopamine is only synthesized in the brain. Norepinephrine is synthesized in the brain and in the adrenal medulla in your body. Then epinephrine is also synthesized in the adrenal medulla from norepinephrine. She did not mention this from the slide, but for your own information, epinephrine is also synthesized in the brain, another version of it. How is epinephrine formed? How is epinephrine synthesized from norepinephrine? Basically, norepinephrine will be released into storage vesicles in the adrenal medulla, and then whenever your body needs to use epinephrine and norepinephrine, or in a fight or flight reaction, those vesicles will be secreted, and norepinephrine will be converted to epinephrine, and then will be secreted into your body, and then they will carry out the physiologic functions. Now, outside the CNS, what does she mean by that? You know how epinephrine and norepinephrine carry out some, let me say, MOL functions or molecular functions, such as, for example, it's you know how epinephrine in the body has a similar job to glucagon. Yes, it inhibits insulin, so it's opposite to insulin. Remember this from previous lectures. As it's written here, they regulate the carbohydrate and lipid metabolism. I'm talking about epinephrine and norepinephrine. Okay, they increase the degradation of glycogen and triacylglycerol. They are opposite to what insulin does. So epinephrine, if you remember the lecture of glycogen, epinephrine has similar job to glucagon in degrading glycogen and using it as an energy source, opposite to insulin. Insulin is used to synthesize glycogen and store it in the liver. So just keep in mind, epinephrine has similar job to glucagon, opposite to insulin. So whenever you're in a fight or flight, this example will make it stick. if you're in a fight or flight situation or fight or flight reaction, you don't want to be storing glucose and storing fat. You want to be using them because most probably you'll be running from a lion, you'll be stressed out on an exam, you'd be doing whatever that requires fight or flight reaction. So you need to use the energy you have in your body. Okay, so when the sympathetic system is working, or epinephrine and norepinephrine are stimulating your physiologic functions in the body, in your bodies, you definitely need the energy from your body. So if you need energy, or I like to think it this way, so it would make more sense for me. So if you're in a fight or flight reaction, or for example a lion is running after you, then you need energy. When you need energy, then you need the glycogen stored in your liver. You need the fat stored in the adipose tissue, and you also need the protein in the muscles, just for the sake of making energy to supply you. Okay, this is how I like to think of fight or flight reactions, because what would happen in my body, or what would happen to this organ, for example, if I am in a fight or flight reaction. That's the question I asked myself to know the answer, basically. طيب, this is just another figure. I will go over it again. I will go over every figure here maybe once or twice, just to make sure that everyone understood everything. So as we said, we start with tyrosine. I know I said we start with phenylalanine, but yes, we do start with phenylalanine, but the main amino acid here is tyrosine. طيب, tyrosine is converted to L-dopa using tyrosine hydroxylase, and tyrosine hydroxylase requires tetrahydrobiopterin, BH4. Then L-dopa is converted to dopamine, and this step is carried out by the decarboxylase enzyme. It requires PLP. What is PLP? PLP is vitamin B5, which is called pyridoxal phosphate. Okay, so the step of converting L-dopa to dopamine requires vitamin B5 or pyridoxal phosphate, PLP. Then dopamine is converted to norepinephrine using the enzyme dopamine beta hydroxylase. Dopamine beta hydroxylase, as I mentioned in the previous two slides ago, requires copper, yep, and ascorbate, which is vitamin C. Yeah, okay. And then norepinephrine will then in the adrenal medulla be transferred, be converted into epinephrine, and this requires a methyl group or a methyl donor, and usually methyl donors in our body is called SAM, basically, because it's S-adenosyl homocysteine, and this is SAM is S-adenosyl methionine. Yes, this is the methionine group. Will be donated to the reaction, or to this enzyme, transferase enzyme, which will give us epinephrine. So let me go over the cofactors real quick here, because they are very important. You will definitely 100% have a question or two, maybe three questions about the cofactors in this in all lectures. Basically, in every cofactor you see, expect a question, at least one question about, because they are the most important things in the whole course, because usually defects of these processes we study are usually deficiencies in these cofactors. For tyrosine, this step of that is carried out by tyrosine hydroxylase requires a tetrahydrobiopterin. It is written here, which is or BH4. But then the step of converting L-dopa to dopamine requires a PLP, a vitamin B5 or pyridoxal phosphate. Then the step of converting dopamine to norepinephrine requires copper and vitamin C, carried out by the enzyme dopamine beta hydroxylase. The norepinephrine is converted to epinephrine carried out by the transferase enzyme, and it requires a methionine donor, which is SAM. Okay. Now, basically everything is written in this slide. Let me go over them quickly, just to make sure we did not miss anything. So tyrosine is first hydroxylated by tyrosine hydroxylase to form L-dopa. Tyrosine hydroxylase requires BH4, which is tetrahydrobiopterin. Okay, you want to see how it's written? It's written here. Tetrahydrobiopterin. This is the rate limiting step of the whole pathway. This is why I said the main amino acid used to synthesize catecholamines is the tyrosine hydroxylase. It is the rate limiting enzyme. Then L-dopa is decarboxylated to form dopamine, and this step requires PLP or vitamin B5 pyridoxal phosphate. Is it vitamin B5 or B6? Let me make sure. 1 second. Okay, it's B6. Oh my God. I've been saying since the beginning of the lecture, it's B5, B5. Okay, it's B6. Sorry for this mistake. PLP is vitamin B6 and it is called pyridoxal phosphate. Yep. Pyridoxal phosphate is vitamin B6, not B5. Keep confusing them. المهم. Can you please check the chat, Mariam? Did anyone mention this to me? Because I've been saying the same mistake since the beginning of the lecture. Let me carry
No,
on.
nobody, just
Again,
right now.
L-dopa is decarboxylated to form dopamine, and this requires PLP or vitamin B6, PLP stands for pyridoxal phosphate. And then dopamine is hydroxylated. Dopamine is hydroxylated. This is the step. Dopamine is hydroxylated into norepinephrine using the enzyme or carried out by the enzyme dopamine hydroxylase to form norepinephrine. And this reaction requires vitamin C, ascorbate, and copper. Finally, epinephrine is formed from norepinephrine in the adrenal medulla. And this requires a methyl donor, which is SAM. Okay. طيب. Let me go over that slide one more time just to make sure you understood everything. Now we have all the information in our heads. Catecholamine synthesis starts with the enzyme tyrosine. It starts with phenylalanine, but the main enzyme here is, the main amino acid is tyrosine. Tyrosine is hydroxylated into L-dopa by the enzyme tyrosine hydroxylase, and this enzyme requires tetrahydrobiopterin in order to form L-dopa. L-dopa is then decarboxylated into dopamine, and this step is carried out by a decarboxylase enzyme. It's called aromatic amino acid decarboxylase, and this step requires vitamin B6, pyridoxal phosphate or PLP. And then dopamine is hydroxylated into norepinephrine, carried out by the enzyme dopamine beta hydroxylase, and this step requires copper and vitamin C. And then in the adrenal medulla, norepinephrine forms epinephrine by the enzyme, by a transferase enzyme. It is called phenylethylamine in methyl transferase. I don't know if I spelled it right, حتى. But the most important piece of information here is that this reaction requires a methyl donor, which is SAM. طيب. I actually don't know why the doctor didn't write vitamin B6 here. She just wrote PLP. Anyways, I'm sorry for the mistake again. طيب. Tetrahydrobiopterin, which is the cofactor that is required by the enzyme the enzyme tyrosine hydroxylase, which converts tyrosine into L-dopa. Tyrosine hydroxylase requires tetrahydrobiopterin. Where does tetrahydrobiopterin or PH4 comes from? It comes from an enzyme called dihydropteridine reductase, or DHPR. Okay. You will study this in the coming after I think 2 or 3 lectures, the lecture called nucleotide metabolism. I don't know if you took this yet or not, but in that lecture you will get explained how this tetrahydrobiopterin comes from, okay, or where does it come from. This slide, the doctor here is trying to say that a deficiency in this enzyme, DHPR, will lead to a deficiency in the cofactor tetrahydrobiopterin. And if we don't have tetrahydrobiopterin, then the enzyme tyrosine hydroxylase will not work, صح? Because tyrosine hydroxylase requires tetrahydrobiopterin. And if we don't have tyrosine hydroxylase, then tyrosine cannot be converted into L-dopa. And if this step can't be carried out, then we cannot form catecholamines generally. Again, if we don't have this cofactor or basically any other cofactor in the whole process, we can't get the final product, which is the catecholamines. طيب. The doctor is focused on this specifically because this is the rate limiting enzyme and because forming tetrahydrobiopterin requires another process by itself, which is carried out by the enzyme DHPR to form the tetrahydrobiopterin. Okay. So she just wrote here what I explained. So in DHPR deficiency, the activities of phenylalanine hydroxylase and tyrosine hydroxylase will be low, or they will not be there. Phenyl hydroxylase, as I said, phenyl hydroxylase is converted to tyrosine. That's the first step. Okay, it's not the rate limiting step though. The rate limiting step is tyrosine hydroxylase. Okay, that's why all our focus is on this step because it's the rate limiting step. However, both of these enzymes, phenylalanine hydroxylase, which converts phenylalanine to tyrosine, and tyrosine hydroxylase, which converts tyrosine into L-DOPA, require tetrahydrobiopterin. Both of them require tetrahydrobiopterin, and tetrahydrobiopterin comes from DHPR or the dihydropteridine reductase. Okay. So if we have a deficiency in this enzyme, we won't have BH4, thus those two enzymes won't be working. We will not have catecholamines. What would we do in that case or in that disease? The doctor here, look at what the doctor here wrote. First of all, okay, let's read this first. Hence, if DHPR is deficient, then we won't have catecholamines because those two enzymes are not working again. Hence, the synthesis of catecholamines and serotonin will be depressed. As I'll be explaining in the coming slides, serotonin is also carried out by the same enzymes, basically, you'll see now. What will be the treatment? First of all, you need to have a diet low in phenylalanine because phenylalanine is a non-essential amino acid and you have to take it from diet. So you have to eat a diet low in phenylalanine to provide and you also have to provide the precursors for catecholamines and serotonin. So you basically have to give L-DOPA because L-DOPA is a precursor of catecholamine, okay, and serotonin as well. So this is L-DOPA. This is what she's basically. This is PKU or phenylketonuria. Parkinson's disease. Parkinson's disease is the disease that the boxer Muhammad Ali Clay had it. It's basically a neurodegenerative disease. The patient will have difficulty moving, will have his posture will look like this. He will have tremors or vibration in his hands, sudden vibrations, slow movement, rigidity, or his muscles will be rigid. He can't move. He or she can't move. His walk won't be normal. If you look at this is called a gait. A gait is an abnormal type of walking. Why or what causes Parkinson's disease? It's basically insufficiency in dopamine production as a result of loss of dopamine producing cells in the brain. Okay, in the brain, I told you dopamine is secreted in the brain, is made and secreted in the brain. If the cells that produce this dopamine are not there or are lost or are died for any certain reason, then you will have Parkinson's disease because you will have insufficient dopamine levels in your brain. طيب. What do we do in this case? Say the same way we do here. What do we give? We give precursors for catecholamines. Dopamine is a catecholamine basically, so you have to give a precursor to it. As I said, it's L-DOPA. You give L-DOPA to Parkinson's patients. The drug is called levodopa. It's the most common treatment. Why don't we, I have a question for you. If we have insufficient, look at this slide to understand. If we have insufficient dopamine in Parkinson's disease, why don't we just give dopamine straight away instead of giving L-DOPA? This is because dopamine cannot cross the blood-brain barrier, okay? Dopamine cannot cross the blood-brain barrier. However, L-DOPA can easily cross it. So we will give L-DOPA. L-DOPA will cross the blood-brain barrier and in the brain will be converted into dopamine. Now, this is a checkpoint. This is a question for you. What is the rate limiting step of catecholamine synthesis? Come on, guys, it does not require
Someone
that
said
much of
A.
thinking. Can I see the
Someone
book?
said A.
Yeah.
B.
No, it's not B, it's A. Excellent.
Most people said A.
Yes, someone said vitamin B6. Thank you, Selma, for saying vitamin B6. Yeah, I was mistaken when I said vitamin B5. But I couldn't see the chat box back then, so yeah, here I am thanking you. المهم, the answer here is tyrosine hydroxylase. It's A. Let me go over the enzymes written over here. So tyrosine hydroxylase is the rate-limiting step of catecholamine synthesis. It converts tyrosine to L-DOPA and it requires tetrahydrobiopterin. Dihydropteridine reductase, or DHPR, is the enzyme that gives us tetrahydrobiopterin, which is the cofactor of tyrosine hydroxylase, and phenylalanine hydroxylase. Phenylalanine hydroxylase converts phenylalanine into tyrosine. Dopamine beta hydroxylase is the step that converts dopamine into norepinephrine. Yep, let me make sure. Yeah, it is the step that converts dopamine into norepinephrine. And it requires vitamin C and magnesium. No, copper. شو magnesium? Copper, yeah, copper. طيب, now since this is a checkpoint, does any one of you have any question of the previous topic I explained? Because this is the most important objective of this whole lecture. No questions? Shall I carry on? No questions. طيب. طيب. Now I will be talking about the degradation of catecholamines. طيب. The degradation of catecholamines is basically, you only need to know those two enzymes. Is carried out by MOL and COMT. MOL is monoamine oxidase, and COMT is catechol O methyltransferase. They degrade catecholamines. Catecholamines are epinephrine, norepinephrine, and dopamine. So both of them degrade all three catecholamines. However, the product of them is different. So the product of the degradation of epinephrine and norepinephrine is vanilmandelic acid, or VMA. And the product of the degradation of dopamine is homovanillic acid, or HVA. Okay, you only just need to know these three pieces of information. Epinephrine and norepinephrine are degraded into vanilmandelic acid, and dopamine is degraded into homovanillic acid. And yeah, both of these products, HVA and VMA, are excreted in urine, and both processes are carried out by monoamine oxidase, or MAO, and COMT, which is catechol O methyltransferase. Right? So if we have a disease that is مثلا a tumor, for example, that secretes a lot of one of the catecholamines, epinephrine or norepinephrine or dopamine مثلا, you will find the respective product increased in the urine. If you tested the urine for a patient who has a tumor that secretes norepinephrine and epinephrine in the adrenal medulla, you will find that the levels of VMA are increased or are so high. Okay? Again, the catecholamines are degraded by oxidative deamination, catalyzed by monoamine oxidase, or MAO, and the second enzyme is methylation, catalyzed by catechol O methyltransferase, or COMT, using a SAM as a methyl donor. So COMT requires a methyl donor, which is SAM. Most of the time it's SAM. There is no order, can occur in any order. Any of both enzymes can work, maybe both of them, no order. Yeah. The products of these reactions are excreted in urine as vanillmandelic acid from epinephrine and norepinephrine, and homovanillic acid, HVA, from dopamine. طيب, I remember we had a question about this last year. Dopamine, she said a patient had this tumor and they tested their blood and they found they tested the urine, sorry, and they found high levels of homovanillic acid. What is increased in their body or in their blood? It's dopamine, or maybe vanillmandelic acid. So it's epinephrine and norepinephrine, because all three are catecholamines. L-dopa is also a catecholamine. Yeah. Oh, yeah. It's written here. This disease, or the tumor in the adrenal medulla that secretes norepinephrine and epinephrine, is called pheochromocytoma. So these products are increased in pheochromocytoma, these two products. It's a rare tumors of adrenal gland characterized by excessive production of catecholamines. It's usually not both products. It's usually vanillmandelic acid, because remember, dopamine was made in the brain. It's only made in the brain. So if someone has pheochromocytoma, it's basically a tumor of the adrenal gland that secretes epinephrine and norepinephrine. So if you tested the urine of a patient who has pheochromocytoma, you will find high vanillmandelic acid levels in the urine. بس. طيب, worth mentioning those drugs or antidepressants. You don't need to focus on them a lot. You just need to know, I think she wrote here too, information. You will take them in much more details in your next year, inshallah. You just, she wrote here again, dopamine and serotonin are degraded by monoamine oxidase. I still didn't explain serotonin, but it's mentioned in it's mentioned here a lot, because as I told you, serotonin shares the same process of degradation and production of dopamine and the catecholamines generally. Monoamine oxidase inhibitors, or MAO inhibitors, are extremely strong class antidepressants. Why? I'll explain why when I finish the slide, please. They treat depression by preventing the breakdown of dopamine and serotonin. They are the first antidepressants developed. They are more potent than SSRIs because they affect more neurotransmitters and they can cause more side effects, like one by one. So but you don't need to know everything, but I like to explain just to make everything clear. So MAO, and once you understand those, that you don't need to know basically the drugs, or maybe a super يعني a disease that she told you don't study. But when you understand it, then 100% you understood the normal physiology, which is basically what's required from you. MAO inhibitors, they prevent so if you inhibit MAO, let me go back. If you inhibit MAO, check out here. If you inhibit MAO, or COMT maybe, but the one that is well known أكتر is MAO, and it's used أكتر is MAO. If you inhibited MAO, then you don't degrade dopamine, then you will have more dopamine in your body, صح? And this is the difference. In depressed patients, they don't have that much of, or they need more dopamine in their and serotonin in their brains يعني. So when you inhibit the enzymes that degrade dopamine and serotonin, you will have more dopamine and serotonin. And that's basically why they use monoamine oxidase inhibitors as antidepressants. SSRIs are selective serotonin receptor inhibitor. So they inhibit, SSRIs inhibit the receptors that take out serotonin. So you will increase serotonin more in the brain and in the blood generally. This way you will increase serotonin and dopamine أكتر. And this is the goal of using those inhibitors or those drugs. طيب, هلا tryptophan is used to synthesize serotonin. We finally explain serotonin now. I think she wrote it here, no, not here. Again, keep in mind that serotonin shares the same steps of production and degradation of dopamine. Serotonin is synthesized from tryptophan. Catecholamines are synthesized from tyrosine. What else is synthesized from tyrosine? thyroid hormone, and melanin, and catecholamines. Okay, don't forget. Tryptophan synthesizes or is used to synthesize serotonin. Tryptophan is hydroxylated by a hydroxylase. Here you go, hydroxylase. What does hydroxylase requires? Tetrahydrobiopterin. 5-hydroxytryptophan is decarboxylated into 5-HT. 5-HT is 5-hydroxytryptamine. 5-HT is basically serotonin. Whenever you see 5-HT anywhere, they are basically trying to tell you that there's serotonin here. So 5-HT is serotonin. Because it will be eventually converted into serotonin using a decarboxylase enzyme. And keep this in mind, any decarboxylase enzyme requires PLP. What is PLP? Vitamin B6, okay? Don't forget, or pyridoxal phosphate. Yeah. And the hydroxylase here requires tetrahydrobiopterin. I want to stress on these because I told you cofactors are the most important things, the whole course, whole MOL يعني. طيب. Sorry. Serotonin or 5-HT, 5-hydroxytryptamine, is synthesized and stored at several sites in the body. They are synthesized and stored in the CNS for all the functions it required in the CNS or central nervous system, and in the intestines. Because serotonin also has many physiological functions in the intestines. So yeah, in the CNS, it functions as a neurotransmitter, like dopamine and norepinephrine and epinephrine in the CNS. They function as neurotransmitters. And remember, catecholamines in the CNS, like serotonin, they function as neurotransmitters. Norepinephrine and epinephrine in the body has physiological functions, the things I explained, where they control the metabolism of carbohydrates and lipids. Serotonin also has multiple physiologic roles, including mood changes, regulation of sleep, appetite, temperature, blood pressure, cognitive functions. All of these in your body are affected by serotonin. What is a clinical aspect of serotonin? Serotonin, or SSRIs, or selective serotonin reuptake inhibitors, they change or they increase serotonin levels, and they are used as antidepressants because, again, depressed people require more serotonin and dopamine levels, so they give them any drug basically that will increase serotonin levels in their brains, such as SSRIs, selective serotonin reuptake inhibitors, where they inhibit the reuptake of serotonin, and MAO inhibitors, which are I forgot, and I won't go back. And again, MAO inhibitors they just inhibit MAO, and MAO degrade dopamine. So we don't want to degrade dopamine. We want it to stay in the brain in order to better the status of a depressed person. Serotonin is also converted to melatonin in the pineal glands, and melatonin is required for sleeping. Once melatonin is secreted in your brain, then it's time for sleep. That's the biological clock in your brains. It's according to the sunlight and according to the day-night shifts in our days, secretes melatonin or inhibits melatonin. If you have more, once the night starts, more melatonin, after sunset, basically melatonin starts to be secreted. The more you proceed with time at night, the more melatonin you will be secreted by the pineal gland that is made from serotonin. And then once you reach to a certain point at night before Fajr, I think it's 2 a.m., 2 or 3 a.m. I forgot. You study it in neuro as well. Melatonin stops being secreted because your biological clock thinks, or that's maybe that's for sure the right system. By then you have to get ready, because after a couple of hours you have to wake up, which is at sunrise. Let's say sunrise is at 5. By 2 or 3 maybe melatonin will stop secretion, and neurotransmitters that stimulate you, and that basically neurotransmitters that stimulate you, and they carry out the wake up function will be secreted more. because after like 2 or 3 hours you are supposed to wake up, but unfortunately this system is we don't adhere to this system nowadays. We don't sleep well, unfortunately. Serotonin is degraded by MAO. I told you, serotonin has similar production steps and the degradation steps as catecholamines by MAO and COMT, and it's produced by a hydroxylase and a decarboxylase. It will be, yes, both dopamine and serotonin are synthesized in a similar way or in a similar pathway, requiring hydroxylation and decarboxylation. The hydroxylation in both processes or pathways require tetrahydrobiopterin, and the decarboxylation in both pathways require PLP or vitamin B6, which is pyridoxal phosphate. طيب, you see here the doctor added this slide for you to compare and not confuse them. Tyrosine for catecholamines, tryptophan for serotonin. Then tyrosine will be hydroxylated using tyrosine hydroxylase to form L-DOPA, and this hydroxylase requires a tetrahydrobiopterin. When compared to serotonin pathway, it starts with tryptophan. It is also hydroxylated, and again, similar to catecholamines, this hydroxylase requires tetrahydrobiopterin. Then L-DOPA is decarboxylated into dopamine. طيب. And this decarboxylase requires PLP or vitamin B6. Similarly here, 5-HT is decarboxylated into serotonin, and this decarboxylase requires vitamin B6 or PLP. This is why they always say dopamine is like serotonin, because synthesizing dopamine and serotonin is very similar. Also, their degradation, both of them are degraded by MAO and COMT. That's why MAO inhibitors increase both levels, dopamine and serotonin. طيب. How is serotonin degraded? I just explained this literally. This is a checkpoint again. If you have any question, just answer this in the chat box and ask me any question if you have. Is it by MAO or monoamine oxidase? Is it by homovanillic acid or methyltransferase or COMT and MAO? Nope, not A. It's D, COMT and MAO. صح? Yeah. Let's go back and check. Oh, okay. The doctor here wrote, it's degraded by MAO only, but from my own knowledge, I know that it is degraded by MAO and COMT. But let's stick to MAO better, عشان because that's what's written in the slides. Although she literally said in the recording, it's done by both COMT and MAO, and from my own knowledge, and Khalid, I know that it's by MAO and COMT, both of them. This is the catecholamines degradation. It's MAO and COMT. Yeah, catecholamines both MAO and COMT. Serotonin, it's only MAO شكلها. طيب, let's stick with اللهم صل على النبي, MAO only. Let's stick with MAO only, since this is what's written in the slide and this is basically the reliable source. طيب, فخلاص, it's MAO. How is serotonin degraded? By MAO. I can ask the doctor and send you on the group on Sunday, inshallah. But for now, stick with MAO, because this is what's written in the slide. Do you have any question? Allah. What is the chat box? Yeah, here we go. Do you have any question, or shall I proceed? No questions. طيب. Histidine. Histidine or L-histidine is an amino acid that is required to synthesize histamine. What is histamine? Histamine is a chemical messenger that is that mediates allergy and inflammatory reactions. You see, whenever you have a runny nose, a headache, or any type of allergy response or allergy symptom, this is always carried out by histamine. That's why whenever you have an allergic reaction, they give you an antihistamine such as Larinase or Zyrtec, for example. Both of them are or inhibits histamine synthesis. طيب, histamine is a chemical messenger that mediates a wide range of cellular responses, including allergic and inflammatory reactions. It also inhibits gastric acid secretion. You took this in GI. Histamine has a role in gastric acid secretion. So if you inhibit it, or one of the histamine receptors has carries out a function in gastric acid secretion. So if you inhibit that receptor, then you will prevent gastric acid secretion. Also vasodilation. Histamine vasodilates. This is what happens in allergies, vasodilation and many other reactions. Let's not talk about them here because they are beyond your scope. Histamine is formed by decarboxylation of histidine in a reaction requiring PLP. I will explain it here now. It is secreted by, okay, I will explain this later. طيب, histidine, we start with histidine. Histidine is the main amino acid that will, it's only one step. Histidine is decarboxylated into histamine. It is carried out by an enzyme called histidine decarboxylase. And this histidine decarboxylase requires the cofactor PLP or vitamin B6, which is called pyridoxal phosphate. طيب, where is it secreted from? Mast cells. So for during trauma or any allergic reaction, for example, if you ate, let's say someone is allergic from peanuts and ate peanuts, mast cells will start secreting histamine. For example, if you had a trauma, you had a cut or anything, mast cells will be one of those immune cells that will be there at the site of trauma in order to secrete histamine, for histamine to start its work from vasodilation and all other functions that let me say reflected into you as the allergic symptoms, which are for example redness, itchiness, runny nose, headaches sometimes. All of those are allergic reactions that are carried out by histamine. And that's why when you take an antihistamine, you will stop all of those allergic reactions. It's written over here. Agents that interfere with the action of histamine have important applications, which are antihistamines. Okay, done with histamine and histidine. Creatine now. Creatine is the source of energy in our muscles. Those who go to the gym and they take creatine, they know that creatine is a huge source of energy. I'll explain to you now how and why. First of all, how is creatine made? It is made from two amino acids, not one, arginine and the glycine. See here, arginine with glycine are forms a product that is called guan. I don't know how to spell it حتى. Guani. The doctor أصلا didn't focus on this, but she literally said arginine and glycine forms creatine eventually, and through two enzymes, an aminotransferase and a methyltransferase. The methyltransferase requires a methyl donor, which is SAM. طيب. She focused on the point of forming creatine or synthesizing creatine requires a methyl donor, which is SAM. Then what's the next step is forming creatine phosphate. Now, what is the difference between creatine phosphate, and creatinine? Now, the active form of creatine is creatine phosphate. So creatine phosphate is synthesized from creatine by the enzyme creatine kinase, or CK. And creatinine is the form of creatine that is excreted. So in order to excrete the excess creatine in your muscles, you need to convert creatine or creatine phosphate into creatinine, and this is the form that will be excreted in urine. From this, you can know or you can extract that if we measured creatinine levels in urine, you can expect, you can know or you can predict the levels of creatine in the body and if they are increased or not. Creatine kinase is also tested in urine, in blood, sorry. And it is indicative of, you must know this, it is in the, because you took it in the lecture of enzymes with Dr. Ahmed. Creatine kinase tells, يعني if you find creatine kinase in blood, this means there's something wrong with the heart. تمام. Okay. This is basically a repetition of what I explained. Let's go over them quickly. Creatine is synthesized in the liver and kidneys from glycine and arginine. It's very important. The whole lecture is about knowing which amino acids form which products. And ASAM. You see this, she actually focused on this, a methyl group and ASAM. Don't worry, at the end I will go back quickly هيك and over. I will not explain anything again, بس unless you want it يعني I'd be more than happy to. بس I will go over it really quickly just to tell you what I expect will be coming in the exam, or if they want to ask you something from this lecture, what will they be asking you about. Like creatine is reversibly phosphorylated to creatine phosphate by creatine kinase using an ATP and a phosphate donor. It's supposed to be written here. Creatine is phosphorylated into reversibly phosphorylated. You have to know this because they can be and it can be reversed by creatine kinase, and this requires ATP. طيب. And a phosphate donor because you need to have phosphate together with the whole phosphorylation is adding a phosphate into to creatine عشان to have creatine phosphate. The presence of creatine kinase, yes, obviously this is the main enzyme أصلا. She said here what I explained إنه CKMB or creatine kinase MB is the isoenzyme in plasma, is indicative of heart damage and is used to diagnose myocardial infarctions. Like creatine phosphate is also called phosphocreatine. It's a derivative of creatine found in muscles. It is a high energy compound. That's why bodybuilders take it as powder, literally a supplement that provides rapid mobilized reserve of high energy phosphates that can reversibly be transferred to ADP to maintain intracellular levels of ATP during the first few minutes of intense muscular contraction. The amount of creatine phosphate in the body is proportional to muscle mass. What is she trying here to say here? إنه creatine is converted to creatine phosphate by the enzyme creatine kinase in order to it is basically the use of energy. That's when you use the energy, that's what happened, because creatine phosphate is a high energy compound, okay? But whenever you want to maintain intracellular levels of ATP, you will convert creatine phosphate into creatine again. That's the usage of it. So you convert creatine into creatine phosphate. In this process, you use energy. طيب, if now I don't have this cell of this literally this molecule of ATP because I use it into forming creatine phosphate. If I want to use this ATP, I will have to convert creatine phosphate again into creatine in order to have ATP. That's why it's a high energy molecule, because it has an phosphate in it. Because what is the difference between ADP and ATP? It's one molecule of phosphate, okay? أصلا from the name adenosine diphosphate and adenosine triphosphate. The difference between ADP and ATP is one phosphate. So if you have ADP and you have a phosphate molecule with it, you will have ATP. And intense muscle contractions requires a lot of ATP. The muscle, when the muscle is under intense exercise or intense contraction, it requires more energy. It will take the creatine phosphate stored and convert it back to creatine, this is how it will have one ATP molecule, or no, I remember from MSK, it has 4 ATP molecules, not one. I don't think she did not mention it here, but I remember it from MSK. And creatine phosphate carries out 4 molecules of ATP. So once your body converts it back to creatine, it supplies 4 اللهم صل على النبي 4 molecules of ATP, and this is why it's a very high energy compound, and this is why bodybuilders use it to supply more energy for them while exercising. And again, this is actually an important piece of information. The amount of creatine phosphate in the body is proportional to the muscle mass, because the bigger the muscle is, the more storage of creatine phosphate it will have, and the more it will be storing creatine phosphate. Why? Because when you need to contract this big muscle, you will require more ATP than contracting a small muscle. For this more ATP, where will it come from? It will come from this creatine phosphate. Logically, the bigger muscle will require more ATP than the smaller muscle, hence more creatine phosphate than the smaller muscle. So more creatine phosphate, more energy. It's a high energy compound. That's it. So you have it. It's worded in a weird way here, but you just have to know creatine phosphate has energy in it. Once you convert it back to creatine, you will have this ATP or energy basically secreted in order for the muscle to use it in its contractions. This has been explained in MSK as far as I remember. How do we degrade creatine? We have to convert it into creatinine, as I said. Creatine and creatine phosphate spontaneously cyclize to form creatinine, which is secreted in the urine. Okay, let me show. Remember this. Look at this. See here how both creatine and creatine phosphate are converted into creatinine. عشان you might have creatine phosphate, you might have excess creatine phosphate stored in the muscle, and there is no contraction, there is no intense exercise. Why would I have excess? Where would I go? I would just convert it into creatinine and excrete it in urine. Maybe you're under intense exercise and you've used all the creatine phosphate in order to form ATP here, carried out by creatine kinase, and then all of this creatine phosphate after will be converted to creatine in order to form this ATP. So we will have excess creatine now. That's why you have a way in order to convert creatine into creatinine, and you don't want it accumulated there. طيب. This is basically it. She also said here, the adult male excretes 1 to 2 grams of creatinine a day. We don't use we have a lot of excess creatine. Creatinine. Okay. And this is you started renal. You will take, if you didn't, I think you took this lecture because it's one of the first lectures in renal. This is a reflection of the kidney function, which is creatinine clearance. Test the urine creatinine clearance, you know the kidney function. طيب. Which is basically the creatinine concentration. This is it. This is the whole lecture. This is the final question the doctor added in at the end. Answer it, please. Creatine phosphate is a critically important. Let me see. There is a question, I guess. A. صح. Thank you very much, Mariam. Let me explain it real quick. Creatine phosphate is critically important molecule for storage of readily available energy in skeletal muscle. The synthesis of creatine phosphate requires the input of which of the following amino acids? It's arginine and glycine. Remember, arginine and glycine, both of them are required to form creatine. It's not asparagine, it's not aspartate, it's not glutamate, it's not glutamine. Okay, this is the lecture. This is the end of it. I just want to go really quick over the slides just to make sure I covered everything. And 1 second. Just to make sure I covered everything and just tell you if there is an important thing. Inshallah, I didn't miss anything. We're almost done. Yeah. Nothing is here important. What is thyroid hormones synthesized from? Tyrosine. Melanin is also synthesized from tyrosine using the enzyme tyrosinase. Remember, albinism is just a defect in tyrosinase. Catecholamines are synthesized from tyrosine as well. 3 enzymes here: tyrosine hydroxylase, decarboxylase to convert L-DOPA to dopamine, hydroxylase again, or dopamine beta hydroxylase, the last enzyme. Tyrosine hydroxylase requires tetrahydrobiopterin. Decarboxylase requires vitamin C and copper. And then the last, dopamine beta hydroxylase requires vitamin C and copper. Decarboxylase requires PLP, vitamin B6, or pyridyl phosphate. Okay, the last enzyme is the transferase that requires a methyl donor. It converts norepinephrine into epinephrine. They are synthesized from tyrosine. Tyrosine. Okay, طيب. Then tetrahydrobiopterin comes from. I think this is very important. طيب. Degradation is carried out by MAO and COMT. And the product of the degradation of epinephrine and norepinephrine is vanillmandelic acid, and the product of the degradation of dopamine is homovanillic acid. Tryptophan or serotonin is synthesized from tryptophan throughout 2 enzymes, hydroxylase and decarboxylase. The hydroxylase requires tetrahydrobiopterin, similar to dopamine, and the decarboxylase enzyme requires PLP, also similar to dopamine, which is vitamin B6. Okay, 5-HT to serotonin, basically. It is degraded by the MAO, as I said, just note for now MAO, and serotonin is a precursor to melatonin, okay, in the pineal gland. Know the difference between dopamine and serotonin and what are the similarities, because I think that's where the questions will come, because it's a place, or it's a slide where students can be confused since they are very similar. The only difference there are 2 more enzymes in forming norepinephrine and epinephrine lately, and they require one of them, which is dopamine beta hydroxylase, requires vitamin C and copper. Okay, vitamin C is also called ascorbate or ascorbic acid. Histidine forms histamine throughout 1 step that is called, and the enzyme is called histidine decarboxylase, which requires PLP or vitamin B6 or pyridoxal phosphate. Here, the notes here written, she's just saying that what happens in an allergic reaction. Yani, read them if you're interested. Or what happens during a trauma, for example. Then creatine is synthesized from arginine and glycine using and it also requires a methyl donor, which is SAM here, in order to form creatine. طيب, يعني from arginine and glycine to creatine, you have to have a methyl donor, which is SAM, and then creatine is converted to creatine phosphate by creatine kinase. Creatine phosphate is a high energy molecule. If the muscle requires energy, it will convert creatine phosphate back to creatine, also throughout the enzyme creatine kinase. This is why it's a reversible reaction, and this is how you form ATP. So from creatine to creatine phosphate, you use the ATP and form ADP. From creatine phosphate here, from creatine phosphate to creatine, you use an ADP to form ATP, okay? And then both creatine and creatine phosphate can be converted into creatinine in order to be excreted in the blood. CK, which is creatine kinase, can tell you, or is an indicative of heart damage if it was found in the plasma. Also, creatinine is reflective of the kidney function. The test is called basically creatinine clearance. You know this by now, inshallah, from renal. Yeah, this is basically it. This is the end of the lecture. If you have any question, please let me know. Where is the box of chat? Yeah. Does any one of you have any question? No questions. طيب, thank you for attending this lecture. And again, if you have any question, don't hesitate to contact me. My contact information is here in the first slide. Thank you very much. See you soon, inshallah. And I hope you all best of luck.